Small changes in sugarcane can sometimes be the first warning sign of a serious bacterial disease. This condition is commonly associated with Xanthomonas albilineans, the bacterium responsible for sugarcane leaf scald. The disease can occur in different forms, including a chronic form and a more severe, rapidly developing form. It can also remain hidden for some time before symptoms become obvious. In the chronic form, the first noticeable signs are long, narrow streaks running parallel to the veins of the leaves. These streaks often look like white pencil marks and may be surrounded by yellow borders. As the disease progresses, the affected tissue becomes necrotic, causing parts of the leaf blade to turn brown and dry out. Infected leaves may initially appear dull or bluish-green before developing brown discoloration and eventually wilting. The disease can also cause stunted shoots and poor plant development. Under environmental stress, particularly during unfavorable growing conditions, young shoots may suddenly die. In mature sugarcane, leaves may begin to dry and die from the tips downward. Lateral shoots can become unusually vigorous, and these shoots may develop characteristic white streaks or peeling tissue. In the most severe form of the disease, mature sugarcane stalks may wilt suddenly. Sometimes there are few obvious internal symptoms, making early detection difficult. The bacterium primarily survives in infected sugarcane stubble and is most commonly introduced into new fields through infected planting material. Mechanical handling is an important pathway for disease transmission. Cutting tools and harvesting equipment can transfer bacteria from infected stalks to healthy cuttings. The pathogen may also survive in certain grasses, including elephant grass, which can potentially serve as alternative hosts and contribute to disease spread. Environmental stress can make symptoms more severe. Drought, waterlogged conditions, and low temperatures may increase the impact of the disease on infected plants. Because the pathogen can be carried in planting material, using healthy cuttings is one of the most important preventive measures. Hot water treatment can help reduce bacterial contamination in sugarcane planting material when performed correctly. One approach is to immerse selected stalks or cuttings in water at approximately 50 degrees Celsius for about 3 hours. However, temperature and treatment duration must be carefully controlled to avoid damaging the planting material. At present, reliable chemical control options for sugarcane leaf scald are limited. Some older recommendations have suggested fungicide or bactericide treatments for cuttings, but their effectiveness and legal status vary by region. Only use products that are officially registered for sugarcane and follow the local label instructions. For long-term protection, plant only disease-free cuttings from reliable sources. Carefully control the movement and exchange of planting material between fields. Clean and disinfect cutting tools and harvesting equipment before moving from infected areas to healthy fields. When selecting planting material, avoid varieties known to be highly susceptible to the disease when resistant or tolerant alternatives are available. Remove severely infected plants and manage grasses or other potential alternative hosts around the production area. Regular field inspections are also important, especially when weather conditions are stressful for the crop. The key symptoms to remember are long, narrow, pencil-like white streaks running along the leaf veins, followed by yellowing, browning, leaf drying, and wilting. In severe cases, young shoots may die and mature sugarcane stalks can wilt suddenly. Using clean planting material, proper sanitation, careful tool management, and appropriate hot water treatment can help reduce the risk of introducing and spreading this disease. Follow the channel for more practical plant care knowledge.